Thanks everybody for coming. I appreciate your time today. My name is Dr. Willie Mays. I'm the co-founder, Executive Vice President of Regenerative Medicine and the Head of Neuroscience at Athersys. There's that. So I want to spend some time today giving you a clinical update on our multi-stem program. So we spent the last 20 years working on this proprietary adult cell type, and we've come to understand a lot about the cells, and we've targeted the critical care segment for use of the cells based on our understanding of all the things the cells do, and more importantly, some of the things the cells don't do. We've worked with an esteemed group of academic collaborators. We've generated over 60 peer-reviewed publications over the last 20 years, and we've come to understand some of the things the cells do, like polarizing microglia in macrophages to an anti-inflammatory standpoint, their effect on Tregs. We've worked in the animal health space with dog and cat and horse versions of the multi-stem product. We have strong intellectual property around this cell product. We've come to understand that the cells do different things depending on where and when you place them, how you get them into the body in the different disease models that we've worked on. And we've even developed some intellectual property and a type of device for storage of the cells under ultra-low temperature conditions at the clinical sites for storing and distributing the cells in the pharmacy. The cells are like type O blood. You thaw the cells, you pull them up in a syringe, you put them into a bag of physiologic saline, and then you get them to the patient where they're administered intravenously. And all of this can be done from the time the floor makes the call in roughly less than an hour. So we currently have 3 open INDs. We have an ongoing phase 2 trial being conducted at a single site at the University of Texas, Houston, called the Matrix trial. We're looking at using the cells to sort of ameliorate the immune dysfunction that takes place after severe hemorrhagic traumas. And then the other 2 programs in ARDS and ischemic stroke, I'm going to be providing some updates today on those 2 programs. So let's start with ARDS. So ARDS is the predominant reason that most of the people died following the COVID-19 pandemic. ARDS is a dysfunction of the immune system in the lungs following infections or trauma or other conditions. We have roughly 200,000 cases a year in the United States, roughly 2.5 million cases globally when you're not having a pandemic. There's no approved treatments for ARDS, and you have roughly a 30 to 50% mortality rate once you're diagnosed with ARDS. And even the people that recover, most patients have to undergo mechanical ventilation to help them breathe, and the patients that survive often have scarring as a result of the prolonged time on the ventilator. So back in the early 2012 timeframe, Dr. Tony Ting and his when he was with Athersys and his group of researchers did a lot of preclinical work, number of different labs, number of different animal models, number of different species, including ex vivo lung work that was done to understand what impact our cells had when administered into animal models of ARDS. And so you have to remember here, you're giving a cell intravenously into an organism that is suffering from a severe lung dysfunction. So if you're giving the cells IV, the first barrier they're going to come upon is the lungs. So we spent a lot of time assuring that there was safety of the cells in these indications. And so what we ended up seeing was that the cells had dramatic impact on reducing edema, improving the outcome and the physiology of the lungs. And they worked through a similar mechanism that we had seen across across a number of our different preclinical models that was basically changing the polarization of the macrophages, decreasing neutrophils, really shifting the immune system from a pro to an anti-inflammatory state. So after we got all the data that Tony and his team put together, we went forward and applied for an IND and ran an ARDS trial long before we ever knew that there was a COVID-19 virus coming down the pipeline. So this was a phase 1 study. It was a safety study to make sure that we weren't going to do harm to the patients. Again, it was a double-blind, placebo-controlled study taking place in both the United States and the United Kingdom. So we had 20 cell-treated patients, 10 placebo-treated patients. And what you see, let's see if I have a laser, back. There, sorry, no laser pointer. What you see is that we improved the median time on the ventilator. Again, it was a safety study, but the primary endpoint was the 28-day ventilator-free readout, and you can see that the multi-stem treated patients had an almost 3-to-1 improvement in ventilator-free days and an improvement in mortality. And if you look at the bottom of the table, you can see the patients that were the sickest, that had the lowest basic pulmonary function, according to the Berlin definition of an ARDS patient, that we're seeing almost a 6-fold improvement in ventilator-free days, and again, mortality improvement. We also, based on some of our animal studies, both in ARDS and in some of the other neurologic indications, we saw that the longer people were evaluated, the longer animals were evaluated after receiving cells compared to placebo, you would see increasing benefit. It did not plateau. The cell-treated animals continued to see benefit. And when we took the ARDS cell-treated patients out and we asked them, how are you feeling at 1 year after treatment, you can see that there's a 2-to-1. 80% of the multi-stem treated patients were feeling like they had an improved quality of life compared to only 40% of the placebo-treated patients. So our partner in Japan, Helios, acquired the rights for the use of the cells in Japan, and they ran a very similar study, a phase 1/2 clinical trial called the One Bridge study. So this was an open label study. It was 30 patients again, 20 of whom received the multi-stem product IV, and 10 of whom just received standard of care. So again, it was an open label study. 28-day primary endpoint, again, was ventilator-free days, and we looked at safety. And upon the conclusion of that, we were in the middle of the pandemic, and they ran a second cohort to evaluate the safety and efficacy of multi-stem in COVID-19 induced ARDS patients. So the take-home message here again is almost a 2-to-1 improvement in ventilator-free days in the multi-stem treated patients in Japan, an improvement in mortality, and again in the COVID patient population there were no safety issues found, and none of the patients who received the cells who had COVID-19 induced ARDS went on to die. So when we pull this data together, well, we can see that when you look at the mean days of improvement, we almost reached statistical significance of p.07 and a 5-day improvement. And when you look at the median, it's basically 124% increased improvement in ventilator-free days across both of these trials. So yesterday we announced, we had a press release letting everybody know that Helios had acquired the global license to develop and commercialize multi-stem for the treatment of ARDS. So some of the specifics in the press release involve we get up to $4.5 million in near-term payments, and then some $150 million in milestones and additional potential royalties as development takes place. Helios has already reached an agreement with the PMDA for the clinical path forward, and as we finalize this agreement and more facts come to bear, we will make more announcements. We also had as part of the press release yesterday, we talked about our stroke program, and I want to spend just a few minutes on this to make sure everybody understands, I think, what the press release really said. So this was data from our phase 2 stroke trial. I've been giving the corporate presentation here at Meeting on the Mesa for the last 5 or 6 years, and so we focused a lot of time going through all the data. So anybody that wants to talk about specifics, you can see me after the session is over. But this data demonstrates the kind of improvement, the continuing improvement you see as a function of cell treatment in the stroke patient population. This is unique. There have been papers that have come out the last couple of years that corroborate that basically when you use an acute stroke treatment, using a 90-day surrogate endpoint as a primary endpoint of efficacy is fundamentally proper, because what you are at day 90 is what you're going to be for the rest of your life for all intents and purposes. So here we're demonstrating that at day 90, although not statistically significant, the cells were having roughly a 10% improvement in patients making a full recovery. But at 1 year, we have basically a 22% improvement that was p less than 0.01. So with this data, and at the end of our phase 2 meeting with the FDA, we reached an agreement on a pivotal phase 3 study under fast track, RMAT, and special protocol assessment to conduct this trial. The primary endpoint for the ongoing Masters-2 trial is modified Rankin shift. So modified Rankin score is a score that doctors give to patients as a global disability score. So 0 means you're normal, 6 means you're dead. The higher the number, the more severely disabled you are. And so what we're looking at is in our pivotal phase 3 studies, we're looking at a patient population of moderate to moderately severe stroke patients, and then we're administering the cells in the 18 to 36 hours after the onset of the stroke. So this is what a modified Rankin shift analysis looks like. This is data from our Masters-1 study. And so you can see what you're looking for, or what you're hoping to see, is that the multi-stem treated groups, you get more of the green color to the left filling up and less of the red and the olive colors to the right. And you can see that the multi-stem treated patients, although not statistically significant at day 90, there's clearly some benefit being provided by the cells. When you look at day 365, you can see that, again, not statistically significant, but was trending in the right direction. When we take a look at, so our partners again, Helios, also have the rights to the cells for treatment of stroke in Japan. They ran a phase 2-3 trial that's very similar to the Masters-2 trial that we're currently conducting. And basically what you see when you look at the shift analysis of the patient population that exactly mimics the type of patient we're hoping to evaluate in Masters 2, you can see that at day 90, again, not statistically significant, but the data, again, the shift analysis improves. You see a shift of improving patients to the left when compared to the placebo-treated patients. So when we got a second trial that demonstrated improving patient outcomes at day 365, we went back to the FDA and said, we asked for a day 365 endpoint as part of our Masters-2 trial. You said no, let's keep it at day 90. We now have 2 trials demonstrating efficacy at day 365 superior to day 90. Can we make a change? They said yes. They agreed to allow us to change the primary endpoint from day 90 to day 365. We said we'd like to do an interim analysis to make sure that our thoughts, our hypothesis, and that the power is still going to be 90%, moving it from day 90 to day 365. So we had an unblinded group at the DSMB of the trial. Once we had 150 patients through day 365, went back and looked at the data and let us know. And the simple take-home was that the 300 patients we were powered for is not sufficient now. We don't understand all of the reasons for this. There could be a lot of reasons. Could be our hypothesis is off. It could be that there's potentially some variability in some of the properties of the cells. It could also mean that we're losing patients to dropout and or mortality as a function of moving it from day 90 to day 365. Regardless, the interim analysis that we announced yesterday was not a futility study, nor did we mean it to be a futility study. We did it to understand how many more patients we need to put into the study to get us powered for success. So we've hired a group of statistical experts. We're going to talk to the FDA to understand what we can and can't continue to look at as a function of this. We don't want to lose the designations that we've received from them. And we're pausing enrollment of the trial until we conduct these analyses. So we'll make more announcements on this in the future. So I just want to say I've spent 20 years working on these cells. There's a group of dedicated scientists and collaborators that we used to have in our European office and people at Athersys, and we spent a lot of time coming to understand what these cells do. And I stand by the fact that these cells are effective in treatment of acute conditions by rebalancing the immune system. So with that, I thank you for your time, and if there's enough time left, I would answer any questions. Thank you.